We now have to move on to an important topic, a very, very relevant high-yield topic, and that's heme synthesis. We're immediately going to follow that up with heme degradation. Really key points, getting late in the lecture series, but let's not lose our energy level. Let's focus on this because this is where the boards really, really want you to understand biochemistry. In heme synthesis, we start out with glycine condensing with succinyl-CoA. This condensation reaction is going to be catalyzed by a molecule called ALA synthase. Okay, the enzyme ALA synthase. What does that stand for? Well, ALA stands for delta aminolevulinic acid. ALA stands for delta aminolevulinic acid. The enzyme that catalyzes its formation is a delta aminolevulinic acid synthase, but to save oxygen, we're going to call that ALA synthase. What's important about this? It's a vitamin B6 requiring enzyme. Why is that important for the boards? Well, there's a certain drug that can actually inhibit vitamin B6. It interferes with vitamin B6 activity. What drug am I thinking of? Well, what drug would you use to treat tuberculosis patients? Okay, hopefully you're thinking of isoniazid. So looking at our figure here, isoniazid therapy inhibits vitamin B6. So it inhibits ALA synthase activity. So oftentimes in tuberculosis patients being treated for isoniazid, people that are TB positive, they're going to get anemias. Now to get around that, you're going to want to supplement these patients with vitamin B6. You're going to want to supplement with B6 to get around the fact that isoniazid inhibits. Okay, the next step in this pathway is the conversion of delta aminolevulinic acid, or ALA, into porphobilinogen. That's catalyzed by an enzyme called ALA dehydrase, or also known as ALA dehydratase. So ALA dehydratase, why is that important? It's inhibited by lead. It's inhibited by heavy metals. Okay, by heavy metal. Heavy metals, not like Led Zeppelin or something like that, but it's inhibited by heavy metals like lead. Lead in particular. So a case could be presented to you where a child's been eating paint chips from the wall, or patients working in a battery factory where they're exposed to a lot of lead, or perhaps they're working with certain pottery glazes that contain lead. That would be a typical situation where you would have a lead poisoning. Okay, moving down the pathway, porphobilinogen can also go through several pathways we're not going to discuss because it's not important, but eventually it's going to be converted into protoporphyrin IX. Protoporphyrin IX is also known as protoheme. Okay, it's protoporphyrin IX. It's the last step before the heme is eventually synthesized. That enzyme is called ferrochelatase. Ferrochelatase, it's another enzyme that's going to be inhibited by heavy metals like lead. It's going to be inhibited by lead, ferrochelatase. What does the enzyme do? It simply takes the iron and incorporates it with the heme molecule. So once again, if lead is a problem, if there's a lead poisoning going on, it's going to inhibit both ALA dehydratase and ferrochelatase, leading to anemia. There's a whole class of diseases of heme biosynthesis, and the boards do want you to be familiar with these to some level. Okay, what I'm talking about are the porphyrias. The porphyrias are a family of diseases where there's a defect in an intermediate step of heme synthesis. So there's a whole host of steps that this figure isn't going into. It's not necessary to know every step, but it is important for you to know some of the porphyrias. The most common one is Porphyria cutanea tarda. There's also another porphyria called acute intermittent porphyria. Okay, both of these porphyrias porphyrias themselves are not very common, but of the porphyrias, these are going to be the two most important ones to remember. Porphyria cutanea tarda, the most common, and then acute intermittent porphyria. Now, what's hallmark for these diseases? What's hallmark is a port wine urine. Okay, a port wine urine. Okay, port wine urine is hallmark for these diseases. So if a patient comes in with purple-colored urine or pinkish-purple colored urine, that's going to lead you towards porphyrias. It's going to lead you toward porphyrias. If we look at our figure here, you'll see that that these diseases are usually autosomal, or for acute intermittent porphyria, it's autosomal dominant. The others are going to be more autosomal recessive. But what you see in these patients can be anxiety, confusion, paranoia. There's actually some kings in the history of England that had some of these diseases. Okay, they would go crazy every once in a while. Well, the reason was because they had a porphyria. It can lead to abdominal pain. Most of the porphyrias involve photosensitivity. So if their patients are exposed to the sun, it can lead to blistering of the skin. But acute intermittent porphyria is a special one. There's no photosensitivity here. No photosensitivity. So one of the caveats here for porphyrias is you don't want to give these patients barbiturates. The barbiturates can cause an exacerbation, actually cause a porphyric crisis to arise. So you want to avoid barbiturate uses in these patients. Let's look at some of the text here for a minute. Look at some of the text. Just to reiterate here, different causes of anemias. Iron deficiency worldwide. This is the most common cause of anemia worldwide. What type of anemia? It's a microcytic and it's hypochromic. Okay, it's a microcytic, meaning the red blood cells are smaller than usual, and it's hypochromic, meaning that the central pallor region of the red blood cells is going to be large. Okay, so when you look at the red blood cells, you see a larger than normal area of central pallor due to the iron deficiency. The other type of anemia here that we discussed is lead poisoning. Lead poisoning, it produces a microcytic, okay, once again a microcytic, but it's a sideroblastic anemia. The red blood cells have ringed sideroblasts in the bone marrow. So that's hallmark for lead poisoning as opposed to the iron deficiency. Now, here we have a table that's comparing the various anemias, because these are all going to be on the same differential. You have to discriminate between the types of anemias. So for vitamin B6 deficiency, remember that's an important coenzyme involved in heme synthesis. ALA synthase is the enzyme, could be inhibited by isoniazid treatment. Either a B6 or an iron deficiency or a lead poisoning is all going to lead to microcytic. It's all going to lead to microcytic. But B6 and iron deficiency, this is going to lead to hypochromic. You're going to get hypochromic anemias here. Whereas for lead poisoning, you're going to get sideroblastic anemias. You're going to get a sideroblastic anemia happening in lead poisoning. Now, in terms of lab values, notice in B6 deficiency, the level of protoporphyrin is going to be decreased. Why is that? It's because in B6 deficiency, you can't even make the protoporphyrin. You can't even make it. Whereas for iron deficiency and lead poisoning, you can make the protoporphyrin, but you can't convert it eventually to heme. For iron deficiency, you can't undergo the last step, the ferrochelatase step where you incorporate the iron. For lead poisoning, you have an inhibition at that same pathway. So in both iron deficiency and lead poisoning, there's going to be an increase in protoporphyrin. Now let's look at delta-aminolevulinic acid. For vitamin B6 deficiency, the levels of ALA are going to be low. Whereas for iron deficiency, they're going to be normal. And then for lead poisoning, they're going to be elevated. A similar results as what you saw for protoporphyrin 9. It's just that for iron deficiency, you can make your delta-aminolevulinic acid, but once again, you can't incorporate the iron molecule. For vitamin B6 deficiency and for lead poisoning, you're going to have an increase in ferritin levels. The ferritin, those are the storage forms of heme, whereas for iron deficiency, ferritin levels are going to be low. And then finally, the iron levels. They're going to be normal for B6 and for lead poisoning, but they're going to be low for iron deficiency. Going to be low. Notice one other thing here, that isoniazid being used treatment for tuberculosis. The isoniazid can complex with B6. That can be a cause of B6. Iron deficiency, worldwide, once again, the most common cause of anemia, iron deficiency. You're simply not getting enough in your diet. Lead poisoning, it's going to be a child eating paint chips, or using pottery glazes, or working in a battery factory, something where people are exposed to a lot of lead. Now we need to focus for just a second here on iron metabolism. Iron metabolism. Because there's going to be diseases, we just saw one, iron deficiency diseases, and then there are going to be diseases where a patient accumulates too much iron. What do we call that? We call that hemochromatosis. Hemochromatosis. So in iron metabolism, we see that typically a person would take in dietary iron. It's going to be in the 3+ form. It's going to be in the 3+ form, and that with adequate amounts of vitamin C, we could convert this into the 2+ form. We could convert it into the 2+ form, so it could be reduced to 2+. In the intestinal mucosa, this iron is going to be absorbed. Now, some of it can be converted to ferritin within the intestinal mucosa. Okay, that would be in the 3+ form and stored. But a lot of it's going to be taken in by the intestinal mucosa and gone into the bloodstream. Now, in the bloodstream, iron doesn't just float around willy-nilly. Okay, what would happen then, if that was the case, is that the iron would turn the blood to rust. Okay, it would get oxidized and you'd start getting rust in your blood. You don't want that. So the iron ends up complexing with a molecule called apotransferrin. When it binds to the iron, we now have transferrin. So transferrin is the transport form of iron. It's the iron transport form. The transferrin will take the iron to other tissues. It could either be stored in the other tissues, or it could be used as for enzymes and cytochromes. Many of your cytochromes are iron containing, so they need that iron in order to function. Thinking about your electron transport chain cytochromes, all those cytochromes, they need some iron. The other place the transferrin can go is to the bone, okay, to the bone marrow specifically. So there's going to be transporters that bring the iron into the bone marrow. It's going to be necessary for erythropoiesis happening in here. Why is that? It's because of the ferrochelatase step. The last step in heme synthesis requires that iron. So when we measure transferrin levels, those are levels of transferrin in the blood. Ferritin is the storage form of iron. And then finally, hemosiderin, that's excess storage form of iron. That stores excess iron, usually within the liver. Okay, that's where most of the hemosiderin is going to be. It's going to be in the liver. So if ferritin's the normal storage form of iron, the form when excess iron beyond just normal storage form, that's called hemosiderin. Notice one other thing about this figure, and that's that the reticuloendothelial system, the cells of the reticuloendothelial system, things like macrophages, especially within the spleen, they can actually break down the hemoglobin. They can break down the hemoglobin, release the iron into the blood, where it can be taken up by the bone marrow specifically. That would be involved in the turnover of these red blood cells that are being made by the bone marrow. So let's now look specifically at hemochromatosis. Very, very likely the boards will ask you a question relative to hemochromatosis. In hemochromatosis, you're going to get hemosiderin deposits in many, many different tissues. Now, what's the genetic problem here? In hemochromatosis, it's thought that the intestinal mucosa, for whatever reason, it's overactive. It's absorbing more iron than it should. This causes iron to accumulate in many of your tissues, leads to problems. So, for example, in the liver iron accumulation is going to lead to cirrhosis. Actually, this is one of the ways in which hemochromatosis is diagnosed. Do a liver punch biopsy, stain the tissues, and you see an accumulation of iron. In the pancreas, the pancreatic tissue is going to be damaged, and this is going to lead to diabetes. In the joints are going to accumulate iron. This could lead to arthritis. And then finally, in the skin, this is another way that hemochromatosis is often diagnosed. The iron accumulates in the skin, and it's going to look like you have a bronze tan, even in the dead of winter. So, hemochromatosis is there's many different manifestations of this. You should be familiar with them. And as far as the blood test goes, the transferrin it typically should be about 1/3 saturated. But in a hemochromatosis patient, this iron binding capacity is going to go way up. It's going to go way up above 50%. So that's indicative of hemochromatosis. That liver core biopsy and staining for iron, that's going to be indicative of hemochromatosis if it's positive. Okay, now we're going to shift gears from heme synthesis and talk about heme degradation. Equally important. Let's take a look here. Most of the heme is going to be broken down in the reticular endothelial cells by macrophages in the spleen. But this can actually happen out in the periphery. Anytime somebody comes along and they're going to punch you and you get a bruise, hopefully it doesn't happen too often, but you get a bruise. The colors of the bruise, what does that come from? It's from the breakdown of your red blood cells. It's from the breakdown of your red blood cells. So it's the heme that's being degraded out in the periphery now. But most of the red blood cells, they go to the spleen to retire. Now, in terms of the heme degradation pathway, heme is first going to be converted to biliverdin, a green-colored pigment. Okay, think of verdant pastures, a green-colored pigment. And then biliverdin is converted to bilirubin. All right, the bilirubin is extremely insoluble. Bilirubin, very insoluble, so when it gets into the bloodstream, it must be conjugated to something. When in doubt, what's it conjugated to? It's conjugated to albumin. So the albumin-bilirubin complex gets delivered to the liver. Okay, this is important, and the albumin drops the bilirubin off at the liver where it undergoes further metabolism. The most important metabolism of bilirubin is shown on the slide right here, and it's the enzyme UDP glucuronal transferase. Remember that enzyme, please. UDP glucuronal transferase. Basically, what happens is this enzyme takes 2 molecules of UDP glucuronate, it transfers them over to bilirubin within the liver, and this creates the molecule bilirubin diglucuronide. Bilirubin diglucuronide is what we know as conjugated bilirubin. Conjugated bilirubin is also known as bilirubin diglucuronide. Important point. Another important point is that bilirubin diglucuronide, or conjugated bilirubin, okay, that's a direct bilirubin. When we measure bilirubins, sometimes they'll say direct bilirubin. Well, that's the conjugated form of bilirubin. The unconjugated form of bilirubin is indirect bilirubin. So keep in mind, unconjugated goes along with indirect. So unconjugated equals indirect bilirubin. So what happens to the bilirubin? After it gets conjugated, it gets actively transported. Into the bile duct from the liver. It then gets into the common duct, enters into the small intestine, and notice that it's going to be further converted to urobilinogen within the small intestine, and of course gets collected by the kidney and excreted in the urine. And in case you ever cared about this fact, it's actually what makes your urine yellow. Okay, under most circumstances, what makes your urine yellow is the heme degradation products. What else can happen to the bilirubin? Well, the bilirubin can be worked upon by your intestinal flora. Your intestinal flora can convert your urobilinogen into stercobilin. It's just another altered form of bilirubin. Stercobilin is a brown-colored pigment. And in case you ever cared, it's what gives feces their characteristic brown color. So the color of your urine and feces, it's from heme degradation. Now, diseases. I don't think the boards are going to ask you what color your urine and feces are, but they will ask you about the diseases here. Okay, we have several. There's going to be, first of all, physiologic jaundice. Physiologic jaundice is when you have low levels of conjugation enzymes in newborns. You know that typically newborns are very susceptible to jaundice. And what's the reason for this? There's a couple. Number one, there's a lot of hemolysis that goes on right after an infant's born. A lot of hemolysis. We have, fetal hemoglobin and a lot of processes, a lot of changes. So there's going to be a lot of hemolysis. That's number one. Number two, the level of UDP glucuronal transferase doesn't really kick in necessarily right away. It takes a little time for this enzyme to get up to snuff, to get up to levels that can adequately handle the bilirubin that's coming into the system. So for that reason, in newborns, we oftentimes see physiologic jaundice. Physiologic jaundice is a defect in UDP not a defect, I shouldn't say. It's caused by a deficiency in UDP glucuronoltransferase, but that deficiency will go away in time. If it doesn't get treated adequately, and how would one treat physiologic jaundice? Well, in the old days, you put your baby in the windowsill, let him have some sunlight, because the sunlight reacts with the bilirubin building up in the skin, and it converts it to a more soluble form. Okay, that's the key with bilirubin, very insoluble compound. Now, in today's world, we would put the baby under a specific light, shining a blue wavelength light on the baby, which helps to, once again, convert that bilirubin into a soluble form. So the diagnostic feature of bilirubin is that jaundice, that yellow color, typically in the skin and in the sclera of the eye. The sclera of the eye. If physiologic jaundice goes untreated in a baby, it's going to lead to kernicterus. Okay, it's going to lead to kernicterus. Kernicterus, if we take a look, is more of a problem because this could lead to neurological deficits in the child. It could lead to cerebral edema and neurological deficits. So kernicterus is a serious problem. The other diseases of heme breakdown, okay, are Crigler-Najjar syndromes and Gilbert's syndromes, sometimes called Gilbert's syndrome. We'll say Gilbert's. But Crigler-Najjar syndrome, what causes that is a complete wipeout in UDP glucuronoltransferase. So, unfortunately, in Crigler-Najjar syndrome, there is no enzyme activity for UDP glucuronoltransferase. As a result, the bilirubin, the insoluble bilirubin, is going to back up. The patients are going to be severely jaundiced. For Gilbert's syndrome, you might not even realize that your patient has this disease until you get out the ophthalmoscope or you closely examine their eye. Okay, you look in their eye grossly. You would perhaps see some sclera, some yellow sclera, and you'd suspect perhaps that this patient was jaundiced, but it might turn out that they just have a slight defect in UDP glucuronoltransferase. So be prepared for the case presentation of somebody with Crigler-Najjar versus Gilbert's. So those are situations that increase indirect bilirubin. Remember, indirect bilirubin is unconjugated bilirubin. If the levels go up, it was most likely because you just couldn't conjugate it. Maybe you couldn't conjugate it fast enough because you had one of these deficiencies, or perhaps you had malaria and your red blood cells were being lysed and there was excess hemolysis going on. So in this case, you're just flooding the system with bilirubin and the liver just can't keep up, okay? It can't conjugate the bilirubin fast enough. So in hemolytic conditions, for example, as we said, malaria, or perhaps a crisis of glucose 6-phosphate dehydrogenase deficiency, a pyruvate kinase deficiency, one of the things you'll oftentimes see in a case presentation is elevated bilirubin levels. And which type? Indirect, unconjugated bilirubin. Next are the diseases where you see increased direct bilirubin. Direct bilirubin. That's going to increase when you have hepatic damage or if you have bile duct obstruction. So the way I want you to think of these diseases where you increase the direct or conjugated bilirubin is when the liver is working, okay, the enzyme activity is working, the UDP glucuronoltransferase, and it's conjugating the bilirubin, but it can't get anywhere, okay? In the case of a bile duct obstruction, it's trying to get into the bile duct, but there's a big stone, like a wall, sitting there preventing it from entering into the bile duct. Or in the case of hepatic damage, the bilirubin's been conjugated, but then the liver cells bust apart and they start spitting the bilirubin out into the conjugated bilirubin out into the blood. So those conditions are indicative of, right, the ones we talked about, hepatic damage, bile duct obstruction. What would indicate those conditions is direct, conjugated bilirubin elevation. Make sure you can discriminate between direct and conditions that increase direct versus conditions that increase indirect. Okay, the hepatic damage, by the way, could arise due to cirrhosis. It could arise due to hepatitis. There's a number of things that could cause the hepatic damage. Finally, there's a couple of other diseases here that are that cause a defect in transporting the bilirubin into the intestine. Remember, this requires active transport to pump this bilirubin into the liver, from the liver into the intestine. This would be Dubin-Johnson's, Rotor syndrome, or obstructive jaundice. Now, in obstructive jaundice, you're going to see these clay-colored stools. Now, why is that? In obstructive jaundice, you can picture there's a big block right here preventing the bilirubin from getting back to the small intestines from the liver, and therefore, the feces are not going to have this characteristic brown color. Instead, they're going to be more clay-colored. They're going to be more clay-colored because the heme degradation product didn't get there. The major causes of jaundice. Let's take a look here. There's 4 major causes of jaundice. The boards are definitely going to test you on this. There could be a hemolytic crisis. As we said, that could be to some type of sickle. For example, somebody goes into sickle cell crisis, right? There's going to be a lot of hemolysis going on. It could be due to malaria. There could be a conjugation deficiency. So this would be either physiologic jaundice, or it could be Crigler-Najjar syndrome, or perhaps Gilbert syndrome. There could be liver damage. That would be cirrhosis or hepatitis. Or finally, there could be bile duct obstruction. So some things that could cause bile duct obstruction, think about a stone there. Think of a patient with cystic fibrosis that's going to have a lot of thick mucus secretions there within the bile duct. Think of somebody with primary sclerosing cholangitis. They're going to have the bile duct basically closing on itself. So bile duct obstruction oftentimes leads to increases in bilirubin. The next thing we're going to discuss briefly is copper metabolism. There's a couple of diseases of copper metabolism that are classically tested on the boards. Okay, I'm talking about Menkes disease and I'm talking about Wilson's disease. The boards want to make sure you understand these 2 diseases. So, intestines, inside the intestines, most of your copper that you obtain from the diet is going to be absorbed. Now, what can happen to this copper? It can be stored. We call the storage form of copper metallothionein. Okay, that's a storage form of copper. Or it can be transported from the intestines. Okay, there's a transporter that transports it from the intestines out into the bloodstream. What are things conjugated to in the bloodstream? Broken record here. They're usually conjugated to albumin. So from the bloodstream, they can get to the liver, and in the liver, we get the copper converted now to ceruloplasmin. This is the active copper carrying protein. So we call it ceruloplasmin. Or on the flip side here, the copper that gets to the liver can be converted once again to metallothionein. Now, once in the liver, the ceruloplasmin can get to the bloodstream. It's going to be important once again for oxidation of iron from the 2+ to the 3+ form. It's going to be important for that step. The excess copper that doesn't get back out into the bloodstream in the form of ceruloplasmin, well, it's going to be excreted in the bile. So that's just an overview of copper metabolism. Now let's take a look at the problems. First of all, you could have a nutritional deficit of copper. If you have a nutritional deficit of copper, the copper levels in the blood and in the liver are going to be low. Okay, you're not getting enough copper in, they're going to be low. Makes sense. Let's contrast that with Menkes disease. Because Menkes disease, you see the same thing. Okay, it's going to look just like a nutritional deficiency. Let's compare that with Menkes disease. Because in Menkes disease, you're going to see a similar situation. There's going to be lower amounts of copper in the blood and in the liver. Okay, so in Menkes disease, I'll tell you where the problem is. The problem's here in the serosal side. You can't get the copper. You're taking in all the copper you want, eating copper pennies left and right if you choose, and the copper can't get from the intestines into the bloodstream. There's a problem with a copper efflux protein. That's the problem in Menkes disease. So that's why it doesn't get into the bloodstream, and it certainly doesn't get into the liver. Now let's look at Wilson's disease. Wilson's disease is more a problem in the liver. So in Wilson's disease, the copper can get into the bloodstream, but it basically gets trapped in the liver. it basically gets trapped in here, and so the ceruloplasmin is going to be lessened. The levels of ceruloplasmin are going to be lower in Wilson's disease, and the levels of ceruloplasmin and copper are going to build up in the liver. Okay, it's going to be low in the blood, high in the liver, and then once again the ceruloplasmin is going to be low out in the blood. So think of Wilson's disease, some of the symptoms with that. Okay, it's going to be neurological impairment, a lot of paranoid behaviors associated with Wilson's disease. What's the pathognomonic feature with Wilson's? Right, you know it. It's the Kaiser-Fleischer rings. The Kaiser-Fleischer rings in the eye, which are caused by the accumulation of that copper pigment. Do you remember the membrane that it accumulates in? They may not say Kaiser-Fleischer rings. They may say Descemet's membrane. Right, Descemet's membrane up there in the eye, the cornea. That's where the copper is going to specifically accumulate. Those are Kaiser-Fleischer rings. So outstanding. That brings us to a close in chapter 17, okay, amino acid synthesis and degradation. I realize this was a very long chapter, but a very important chapter. I recommend that prior to you taking your exam, you come back and review this chapter again, because it's really going to pay dividends for you on your exam. Okay, I have some good news for you. The good news is that we've reached the final chapter of our biochemistry review, Purine and Pyrimidine Metabolism. Some of the most dreaded biochemistry of all medical students. Okay, let's break it down to its most essential parts, and we're only going to hit the points that the boards want you to know. Okay, the first thing we're going to look at is nucleotide synthesis. There's salvage pathways by which nucleotides are pretty much recycled. Okay, you have a good recycling program going on for nucleotides. And then there's a pathway by which we can make the nucleotides from scratch if we need to. We call that the de novo pathway. Okay, let's look at our slide. And on our slide, we see that via the hexose monophosphate shunt, remember the stepchild of the glycolytic pathway, the ribose 5-phosphate could be produced. There were two things produced in that pathway, NADPH and ribose 5-phosphate. Now, the ribose 5-phosphate that gets produced in the hexose monophosphate shunt is going to be the starting point for synthesizing your pyrimidines and your purines. So the ribose 5-phosphate, it gets converted to an intermediate here that's called phosphoribosyl pyrophosphate. That's called PRPP for short, phosphoribosyl pyrophosphate. So this is basically the ribose 5-phosphate that's been activated by having a pyrophosphate moiety added to it. This is then going to be used for the de novo synthesis of your pyrimidines, necessary for the nucleotide synthesis in DNA and RNA. But also, phosphoribosyl pyrophosphate is an important starting point from nucleotides that are going to be recycled. So purines and pyrimidines can also enter into this pathway via a salvage pathway. So the idea is that there's a salvage pathway and a de novo synthesis pathway that can lead to nucleotide synthesis. Most of the problems we're going to see is in the purine salvage pathway. That's going to be the really important one to remember. All right, we'll start out with pyrimidine synthesis. In pyrimidine synthesis, you have a similar situation to what we saw for the urea cycle. Okay, you have a carbon dioxide molecule coming together with a glutamine. There's a molecule generated called carbamyl phosphate. Does it sound familiar? Well, it should. But the key difference here, there's two key differences. Number one is that this is happening out in the cytoplasm. The first step of the urea cycle, remember, happened within the mitochondrial matrix. The second key point is that the enzyme that's involved here is called carbamyl phosphate synthetase 2. Okay, carbamyl phosphate synthetase 2. Whereas for the urea cycle, it was carbamyl phosphate synthetase 1. Why is that important? It's important, first of all, the goal of this entire pathway is to make DNA. Let's not forget that. So why is this first point I mentioned important? It's important for ornithine transcarbamylase deficiency. In ornithine transcarbamylase deficiency, remember, you had an accumulation of carbamyl phosphate within the mitochondria, and that carbamyl phosphate could leak out of the mitochondria, and it now becomes a substrate for pyrimidine synthesis. So what ends up going on is that your body is going to accumulate orotic acid, and it's actually going to accumulate uracil intermediates. So that's why for ornithine transcarbamylase deficiency, we see orotic aciduria and you see increases in uracil. We didn't see that for, remember, carbamyl phosphate synthetase deficiency. So other than the fact that orotic acid is within this pathway, I wouldn't worry about any of these intermediate steps. What I would worry about is this enzyme called ribonucleotide reductase. Ribonucleotide reductase is what takes a ribonucleotide and converts it to a deoxyribonucleotide. So ribonucleotide reductase is what removes that hydroxyl group from the 2' carbon of the ribose sugar. It gets us from being something that can go into into something that can be going into RNA to something that can go into DNA. Why is this important? It's because hydroxyurea is a drug. Hydroxyurea can block ribonucleotide reductase. Okay, that's a drug that's used to treat, of course, cancer and certain dermatologic conditions, so hydroxyurea. The next enzyme in this pathway, thymidylate synthase. Thymidylate synthase takes DUMP, right, DUMP, deoxyuridine monophosphate, or DUMP, DUMP, and converts it to deoxythymidine monophosphate, or DTMP. So if you remember, U's are found in RNA, T's are found in DNA. So this enzyme thymidylate synthase has a net effect of converting U's to T's. Very, very important for DNA synthesis. What's the drug that blocks this? Well, it's the student's favorite drug. It's 5-FU, 5-fluorouracil, right? I won't give you a symbol for it, but 5-FU or 5-fluorouracil is the name of the drug that will block thymidylate synthase. Once again, this could be very useful for treating cancer, for certain dermatologic conditions, 5-fluorouracil. Another enzyme that goes kind of in conjunction with thymidylate synthase is dihydrofolate reductase. So dihydrofolate reductase, if you remember, back to the folic acid synthesis pathway, dihydrofolate reductase is necessary for taking dietary folic acid and converting it to the dihydrofolate and then the tetrahydrofolate form. Okay, so these are varying forms of folic acid. This recycling is absolutely necessary for folic acid to work as a cofactor for methyl donor reactions. Remember, folic acid is an important methyl donor. So in order to convert uracil to thymine, remember there was just a little methyl group on thymine that wasn't present on uracil. Okay, that's going to require folic acid. It requires the enzyme thymidylate synthase and the enzyme that we're showing here, dihydrofolate reductase. What's clinically important? Well, it's clinically important that this enzyme activity can be blocked through certain drugs. So methotrexate. Methotrexate, you know that's used as a anti-cancer drug. It's used to treat rheumatoid arthritis. So methotrexate is an important drug, and it blocks dihydrofolate reductase. How does that help? Well, how that helps is, in the case of cancer, is you prevent the synthesis of DNA because the nucleotides aren't going to be there to make the DNA. And in the case of rheumatoid arthritis, you're going to prevent the rapid turnover of your immune cells that would be attacking your joints. So methotrexate, an important drug. Trimethoprim. Trimethoprim is going to specifically block prokaryotic dihydrofolate reductase, whereas pyrimethamine is going to block protozoal dihydrofolate reductase. So these are going to be useful as antibiotics. Okay, let's take a look at a table that's going to compare these deficiencies with one another, because extremely likely they could give you a patient with rheumatoid arthritis or a cancer patient, say that they're going to be treated with some sort of chemotherapeutic agent, and they want you to know the mechanism behind that particular drug. So that's what this table is comparing. It's comparing the activities and functions of ribonucleotide reductase, thymidylate synthase, and dihydrofolate reductase, and then telling you which drugs can block these enzyme activities. So ribonucleotide reductase, its job is to take all NTPs, nucleotide triphosphates, and reduce them to deoxynucleotide triphosphates. This is necessary for making DNA. What's it blocked by? Hydroxyurea. Thymidylate synthase. Thymidylate synthase is an important enzyme. It's what methylates DUMP, converts it to DTMP, and it requires tetrahydrofolate. Okay, important to remember that this requires tetrahydrofolate. This is going to be blocked by 5-fluorouracil. Finally, dihydrofolate reductase, the job of this enzyme is going to be to convert, not listed here, but to convert folic acid to dihydrofolate and then dihydrofolate to tetrahydrofolate. If you don't have dihydrofolate reductase, you're going to stop DNA synthesis. And why is that? It's because that thymidylate synthase requires the folic acid. It's going to be used in eukaryotes. You're going to inhibit this enzyme activity via methotrexate activity. In protozoans, you're going to use pyrimethamine to inhibit this enzyme activity. And then in bacteria, you're going to use trimethoprim to inhibit this activity. Now that brings up an interesting point, which is illustrated here, and that's cotrimazole therapy. In utilizing cotrimazole therapy, you're basically combining sulfamethoxazole and trimethoprim. So many times for an antibiotic, trimethoprim, sulfamethoxazole will be given in conjunction with one another in one formulation. Now, how would this work? The sulfa drugs, first of all, they're going to block the conversion of para-aminobenzoic acid to folic acid. So they block the conversion of PABA to folic acid. This doesn't cause a problem for us as people because we don't have an ability to make our own folic acid. Remember, we have to get it in through our diet. So the bacteria, they have to take PABA and convert it to folic acid. If sulfamethoxazole therapy is initiated, it's going to block the bacteria from making their own folic acid. And then using this in conjunction with the trimethoprim is going to inhibit dihydrofolate reductase activity. So if you prevent the bacteria from making their own folic acid, and then on top of that, you prevent it from recycling its folic acid to a useful form, it's a very potent way of treating a bacterial infection. So it's commonly used therapy, the cotrimazole therapy. Here's a figure just showing you ribonucleotide reductase, just emphasizing that it's the enzyme that converts all four here of your nucleotide triphosphates into deoxynucleotide triphosphates. Then there's an enzyme called thymidylate synthase. It's necessary for converting DUMP to DTMP. Let's review what drug blocks